Well, today we will discussing about the next part in the paper under the embryology, that is the embryo sac, and the types of embryo sac or classification of the embryo sac. This is most important. हा एक तर 10 मार्काला प्रश्न येतो किंवा एखादा टाईप जो आहे याच्यातला तो 5 मार्क्ससाठी येत असतो. त्यामुळं एवढं दिलेलं आहे तेवढं पुरेसं आहे. Very finely we observe all these things very nicely. What is meant by embryo sac? Embryo sac is nothing but a female gametophyte which develops after fertilization of the flower within the ovule. It is part of embryology or megasporogenesis, the last step in the megasporogenesis, commonly found in the angiosperms. And what it concerns about their structure, the structure is generally in the angiosperm is 8 nucleated structure, and most of the cases 80% or 70 to 80% angiospermic plant shows such type of the 8 nucleated embryo sac, and firstly observed in the genus Polygonum, so it is called as a Polygonum type of the embryo sac. And what about classification or types? It has been concerns that classify this embryo sac depending upon the megaspore nuclei which are participating in the development of the embryo sac. So first about we look at here the monosporic, bisporic and tetrasporic. What about the monosporic? Monosporic means the single cells of megaspore has been take part in the development of the female gametophyte of embryo. We look at here. These are 4 megaspores, and out of these 4 megaspores, megaspore mother cells then divides to form dyad to form the tetrad, and out of these tetrads, one chaliceal end cells has to be functional, and this remaining 3 cells micropylar ends becomes as degenerated. These functional cells first divides to form 2 nucleated cells, 2 to 4 nucleated cells, and finally 8 nucleated cells. These 2 nucleated cells then firstly migrated towards the 2 poles. We look at here in the next colored diagram. Then each of them has to divide to form 4, 4 to 8, and reorganized at 3 plus 2 plus 3, and this is the monosporic type of the embryo sac. This monosporic type of the embryo sac firstly studied in Polygonum by Strasburger in 1879, so it is named as Polygonum type of the embryo sac. The second one is bisporic. Bi means 2. Only 2 megaspore cells has been takes place in the formation of the development of the embryo sac, so it is called as a bisporic. We look at here in this structure 2 cells dyad. From this dyad it has been then divides and forms 4 nucleated cells, 4 to the 8 nucleated cells, and 8 nucleated cells reorganized just as 3 plus 2 plus 3, same as that of the first one, that is the monosporic type. But what is the difference between both of them? Only they are the after the dyad of the megaspore mother cells has been take part both of the 2 cells in the development of the embryo sac development, and it is first time discovered in the Allium fistulosum by Strasburger in 1879, so it is called as also a Allium type of the embryo sac. Second one, finally, tetrasporic embryo sac. This is also called as a peperomia type because of Campbell and Johnson reported this type of the embryo sac in the Peperomia placida in 1900. In this case, what happened? Megaspore mother cells divides to form 4 haploid nuclei, and these 4 haploid nuclei that are not separated from each other or by the wall, so it is likely as words or functions and regarded as megaspore nuclei, and these nuclei divides twice to form 16 nucleated structure, and this 16 nucleated structure organized in this way, that is the two lies at micropolar end. One of them is egg and one is synergis. 8 nuclei at the center forming secondary nucleus, and 6 lie at chalazal end forming the antipodals. That means here the very different structures has been discussed about the tetrasporic embryo sac. इथं चुकलेलं आहे बरं का, 2 अधिक 8 अधिक 6 पाहिजे, इथं तर 5 झालेलं आहे. व्हेरी सॉरी. So note down this part only. It is 2 plus 8 plus 6, that is 8 nucleated embryo sac called as a tetrasporic, and it is called as a peperomia type. So this is about the 3 important types of the embryo sac. Thank you.